So first, what is epigenetics? Epigenetics refers to heritable changes in gene activity and expression that do not involve alterations to the underlying DNA sequence. It plays a crucial role in regulating various cellular processes, development, and adaptation to environmental factors. Unlike genetics, which focuses on the DNA sequence itself, epigenetics delves into modifications that influence how genes are turned on or off. Next, let's move on to the basics of epigenetics. Central to the field of epigenetics are three primary mechanisms: DNA methylation, histone modification, and non coding RNA. DNA methylation involves the addition of methyl groups to cytosine bases, often resulting in the silencing of gene expression. This process is pivotal in embryonic development, genomic imprinting, and X chromosome inactivation. Next, histone modification revolves around altering the structure of histone proteins around which DNA is wound. Modifications such as acetylation, methylation, phosphorylation, and ubiquitination can influence chromatin structure and consequently gene expression. Last is non-coding RNA, including microRNAs and long non-coding RNAs, play a role in regulating gene expression by either inhibiting translation or degrading mRNA. These epigenetic mechanisms collectively contribute to the intricate orchestration of cellular processes, development, and responses to environmental cues, setting epigenetics apart from the conventional focus on DNA sequence in genetics. Next, let's move on to epigenetics and its relationship to disease development. First is autoimmune diseases. Autoimmune diseases are characterized by aberrant DNA methylation and histone modifications that have been linked to the dysregulation of immune responses. For instance, in rheumatoid arthritis, there is evidence of DNA hypomethylation in the regulatory regions of immune-related genes, contributing to the autoimmune pathology. Next, cancer exhibits epigenetic alterations as hallmark features. DNA hypermethylation, particularly in the promoters of tumor suppressor genes, and histone modifications play a crucial role in promoting uncontrolled cell growth. A notable example is the promoter hypermethylation of the BRCA1 gene in breast cancer, illustrating the impact of epigenetic changes in cancer initiation and progression. Then we have neurological problems. Neurological problems are also influenced by epigenetic modifications, which play a pivotal role in neurodevelopment and synaptic plasticity. Aberrations in DNA methylation, for instance, have been linked to neurodegenerative diseases like Alzheimer's and Parkinson's. Altered DNA methylation in the promoter regions of genes associated with Alzheimer's disease exemplifies how epigenetic dysregulation contributes to neurological disorders.
So let's proceed to the epigenetic mechanisms. So epigenetic mechanisms are like control switches in our cells, guiding them to become specific cell types. And there are six mechanisms, and we will be discussing them briefly one by one. So now let's discuss the DNA methylation or the genetic tag. So think of it as putting tags on certain genes. So these tags tells the cell which genes to use and which to ignore during the development. So DNA methylation is a process where a methyl group is added to a specific cytosines in the DNA sequence, primarily at the CPG sites. So these modifications is catalyzed by enzymes which is called the DNA methyl transferases, which results in 5-methyl cytosines. So now let's discuss the histone modification or the cellular architects. So imagine architects arranging blocks, which is the histones, to build a structure, which is the chromatin. So modification to these blocks control how accessible genes are. So some modification open genes for use while others close them. Histone modification involves chemical change to histone proteins, shaping chromatin structure and influencing gene expression. So let's define first these two terms, the condensed state and decondensed state. So condensed state is chromatin is tightly packed, limiting the DNA accessibility and often associated with gene silencing. On the other hand, the decondensed state, the chromatin is more relaxed, allowing greater DNA access and promoting gene expression. So there are several key modifications, and this includes the acetylation, which it opens the chromatin, the methylation, which is the context-dependent activation or repression, the phosphorylation, which regulates the chromatin compaction, and lastly is the ubiquitation, which affects gene expression and cellular processes. So the dynamic interplay of these modifications creates a histone code which conveys information that is crucial for epigenetic regulation and cellular functions. So now let's discuss the chromatin remodeling. So this is a dynamic process of modifying the structure of the chromatin, the combination of DNA and histone proteins. So this remodeling involves the repositioning or modification of nucleosomes. So this is the basic unit of chromatin. By rearranging nucleosomes, cells can have control the accessibility of DNA that influence gene expression. And this chromatin remodeling is crucial aspect of epigenetic regulation because it allows cells to dynamically regulate various cellular processes such as transcription, DNA repair, and replication. So now let's discuss the epigenetic memory. So it's like a cell's identity card. Epigenetic memory ensures that cells remember who they are and pass that information to their daughter cell during cell division. So epigenetic memory refers to inherited modification to cell's DNA which can alter gene expression and cell properties and behavior. So lastly is the cellular plasticity or adaptability. So cells can adapt to changes in their environment. Epigenetic changes allow cells to switch roles or respond to signals providing flexibility. The aforementioned mechanisms ensures that cells plays its important parts and maintains its unique identity in the grand performance of life.
And now let's talk about the fourth objective, which identifies the gaps and issues that challenge the theory of epigenetics on the mechanisms of development and pathology in the human body. So first, we have incomplete understanding. Despite significant progress, our understanding of epigenetic mechanisms is still incomplete. There are many aspects of epigenetics that are not yet fully understood, including the precise mechanisms by which epigenetic modifications occur, as well as their functional consequences. Second would be complexity and variability. Epigenetic processes are highly complex and variable. The same epigenetic modification can have different effects in different individuals or contexts, making it challenging to establish clear cause and effect relationships. Third is the environmental influence. Epigenetic modifications are influenced by environmental factors such as diet, stress, and or exposure to toxins. However, the extent to which these environmental factors contribute to epigenetic changes and their long-term effects on development and pathology are still being explored to this day. Fourth, we have transgenerational inheritance. The concept of transgenerational epigenetic inheritance, where epigenetic modifications can be passed on from one generation to another, is still controversial to this day. While some studies suggest the possibility of transgenerational epigenetic inheritance, more research is needed to understand the mechanisms as well as the implications of this phenomenon. Fifth is epigenetic plasticity. Epigenetic modifications are known to be reversible and can be influenced by various factors throughout an individual's lifespan. However, the extent and limitations of epigenetic plasticity are not yet fully understood. Sixth are the health and social implications. Epigenetic mechanisms challenge the notion that traits present at birth are solely determined by genetics. This raises questions about the distribution of traits and challenges the premise of fairness in health justice theories such as the Rawlsian fair equality of opportunity. Seventh is the biosocial perspective. Epigenetics aims to understand the complex interactions between biology and the social and material environments. However, some research on the development of epigenetic biomarkers tends to focus more on biological determinants of health, neglecting the social and environmental context source. Eight is post-genomic discoveries. The centrality of genes in explaining disease and developments has been challenged by post-genomic findings from disciplines like environmental epigenetics, microbiomics, and developmental origins of health and disease. Influence on evolution. Epigenetics plays a significant role in gene regulation, phenotypic plasticity, development, and the preservation of genome integrity. Understanding how epigenetics influences the course of evolution is an ongoing challenge. And last but not the least are ethical considerations. Epigenetic research, particularly in the context of DNA methylation, raises ethical issues. DNA methylation can regulate gene expression and alter gene function, which has implications for understanding disease as well as human traits.